Hello, hello everyone. I hope everyone can hear me well. Uh, Thank you very much, first of all, for joining us for the BioLevate seminar of today. Today, we are very happy to host Dr. Sagar Arya, which he is a biochemist and interdisciplinary researcher specializing in antimicrobial technology, bacterial membrane system, and nanobiotechnology. He completed his PhD in biochemistry at Deakin University, Australia, where his research focused on bioengineering of specialized metabolism and developing innovative methodologies for reversing bacterial resistance using nanomaterials. Following his doctorate, he pursued postdoctoral research at Khalifa University, where he continued advancing bacterial membrane-based platform for biomedical applications. He later joined Central European Institute of Technology in the Czech Republic, contributing to the development of biohybrid nanomotors designed to capture and neutralize pathogenic bacteria. Dr. Sagriara is currently an MSc CA fellow at Czech Advanced Technology and Research Institute at University of Olomouc. His research focused on the development of a flying seed-inspired sensor for remote environmental monitoring, as well as bacterial membrane-derived vesicles designed to enhance plant immunization in inhabited environments. His work investigates molecular biology, nanotechnology, and applied microbiology with the broader objective of addressing global challenges related to antibiotic resistance, infectious diseases management, and environmental sustainability. Today, we are very happy to host Dr. Sayed. Thank you very much, Dr. Sayed, for being with us today. And he's going to give us a talk titled Nanoscale Bioelectronics and Biohybrids to Tackle Antimicrobial Resistance. Just a note before we jump into the presentation, we are going to have all of your questions by the end of the seminar. So if you have any questions, please write them down in the Q&A box, or you can raise your hand at the end of the talk, and we are going to respond by the end of the talk. And with no further delays, I would say, Sergio, please, the floor is yours. You can start sharing your slide and start the seminar.
Sure. Thank you so much, Latifa. Ciao, everyone. Thank you very much, Latifa, for the kind introduction and also for introducing me to this BioElevate seminar series and for the opportunity to present nanoscale bioelectronics and biohybrids to tackle antimicrobial resistance. I hope I am audible, right?
Yes, we can hear you.
Perfect. Okay, so before going into the details, let's see some background on antibiotics, bacteria, how this antimicrobial resistance develop and what it is leading to in near future. So I hope most of us has been contracted with bacterial infection at least once in our life, and we have been taking antibiotics to deal with the bacterial infection. Now, these antibiotics act on certain vital mechanisms of bacteria, like DNA, RNA, or protein synthesis mechanisms, or certain vital components of bacteria, like bacterial enzymes, efflux pumps, ionic transport mechanisms, membranes, cell wall, so on and so forth. So when we take antibiotics, usually the response is bacteria dies. But when bacteria thinks that something is killing them, they start developing certain resistance mechanisms. Now, these mechanisms include some enzymes, some efflux pumps, or also modification into the cell membrane of the bacteria. So what happens is these enzymes either degrade antibiotic or efflux pumps throw out the antibiotics from the bacteria, or modification in the cell membrane does not really allow the antibiotics to go inside the bacterial cell. So altogether, these are few examples of bacterial resistance mechanisms. Now, what is happening is the rate at which bacteria is developing resistance against these antibiotics is more compared to the rate at which new antibiotics are coming into the market. Now, this is drying up the pipeline of antibiotics to bacterial resistance ratio. So this is classically called as the antibiotic discovery void. That is, for example, you can clearly see from 1987, there hasn't been any new novel antibiotic being discovered or brought into the market. So what we really rely on is 35-year-old antibiotics or modifications of the older antibiotics to treat modern bacterial infections, which doesn't really sound good, right? So what is happening is this is leading to overuse of the old antibiotics. And this overuse has led to the emergence of antimicrobial resistance by the bacteria. So one classical example of this antimicrobial resistance is, I've taken it from 2017, wherein a woman from Las Vegas, Nevada was infected by a bacteria called as a superbug because this bacteria could not be treated with even 26 different antibiotics available in that hospital at that moment of time. So you can imagine the severity of antimicrobial resistance, which is growing gradually and has now turned into a silent pandemic, which is running parallel to our worlds, but people are not aware of it. And this, if it is not uh, dealt with properly, is going to lead to a future wherein we can see more AMR related deaths compared to that of all the cancer related deaths combined together by the year 2050. Now, to not have such type of future, all the global health organizations have come up with certain recommendations for the use of modern technologies. So of these recommendations, I have chosen four different ones because I've been working on uh, the the things which adhere to these four recommendations. Now, the first one of this is developing physiologically relevant technologies which really mimic the growth and treatment of bacteria inside a human body. Then the second one is we need rapid screening platforms and discovery platforms for new antibiotics, which is going to fill up this discovery void. Then what we need is reducing the overuse of older antibiotics, because if we overuse them, then there'll be rise in antibiotic resistance. And the fourth one is replacing the use of antibiotics with alternative natural strategies to overcome the problem of AMR. So these are the four recommendations, of which two of the first recommendations I've been working on relates to bioelectronic platforms. And the last two recommendations given by WHO, I've been dealing with biohybrid platforms. So my subsequent presentation will be divided into four different chapters. Two will be of bioelectronic platforms for testing and developing antibiotics, as well as two will be divided into biohybrid platforms based on reducing the dependence of antibiotic or or completely replacing the use of antibiotics. So to begin with, uh, we we usually get infected with bacterial infections wherein the bacteria is not growing as a single cell in our body. So the bacteria infects by growing in the form of communities wherein they develop bacterial biofilms and layers, which happens in 25 to 80% of the cases, which turn chronic. And these chronic infections are highly resistant to antibiotics as well as immune responses. Now, to treat these infections, what the doctor prescribes antibiotics are based on static tests, which are traditional and considered as standard for now. But what is happening is this test doesn't really produce the dynamic flow environment of the human body. That is the dynamic flow environment, which means our blood is constantly flowing. There is a dynamics between the ionic condition of different parts of our body. Then there is a dynamic condition in the urinary system of our body. So these statics test doesn't really reflect the actual condition of the body, which leads to the treatment failure as well as accelerates the development of antibiotic resistance even further. So what we really need is replacement to this static test with more biologically relevant platforms. So to do and deal with this, we have come up with a microfluidic platform which provides a base for bacterial growth as well as treatment regimes, and that too happens in real time by monitoring it in the form of electrical signals or optical microscopy. So what we essentially did is we took a PDMS body having a wavy microchannel, which mimics the physiologically relevant design of the blood vessels or the urinary tract system. Then this architecture can be combined in a modular fashion in a Lego-like manner that you can increase or decrease the size of this. Then even you can change the shear stress, which is there in the blood vessels, as well as it can be combined with multimodal monitoring system through electrical monitoring as well as optical monitoring. So to achieve this, what we did, we also added P.PSS layer on the top of gold electrode, making it more biocompatible, as well as since P.PSS can directly transduce ionic fluctuations into the biofilm microenvironment and transfer it into an equally important selective and sensitive electrical signal. And then its oxidative state also helps as electron acceptor, wherein it facilitates proper respiration of bacterial biofilm and its growth. So this is the model that we prepared. You can see this was the chip with the electrodes. So what we initially did, we had to run some fundamental studies wherein we wanted to see if the bacteria is able to attach or grow further. We took Pseudomonas aeruginosa, which is one of the critical priority pathogen listed by WHO. So we let the cells grow and attach for a period of 2 hours. Then over a period of 12 hours, the cell grew into microcolonies, and in subsequent 24 hours, the biofilm matured, forming a dense multi-layered structure throughout the surface of the chip. And then in this forms an optimal timeline, which actually happens in the human body, wherein the biofilm matures in the human body, and then we feel that we are being infected, and we need to go to the doctor for getting the antibiotic. So this became optimal time point for us to start the treatment with the antibiotic. So what we did in the first chip, we let this happen for 24 hours. Then we attached a second chip in a Lego-like manner and flushed in the antibiotic for next 24 hours. Now, what we saw is in the second 24 hours, when the antibiotic was flushed in, the bacteria could not grow on the second chip. You can see the migration was very less and the antibiotic treatment was effective. But when the antibiotic treatment was stopped for the subsequent 24 hours, the bacteria were able to migrate and form colonies on the second chip, which clearly demonstrate the high risk of infection recurrence and spread from incompletely eradicated film. So if this thing is being done in static traditional assay, this amount of antibiotic is sufficient enough to kill all the bacteria. But this does not really reflect in dynamic environment. And that is why there is a need to have a dynamic platform, a physiologically relevant platform to test the growth of bacteria as well as test different regimes of antibiotics. So all in all, this system altogether is robust, scalable, and biologically relevant, and could be at some point be replaceable for traditional methods. And then it is possible to have modularity, optical visualization, electrical sensing, as well as making it more physiologically relevant, since it can be scaled up based on the experimental setup as well as the fundamental research based on as well as the fundamental requirements of the research setup. So if you want to go in more details of this work, you can scan the QR code and go through the article. If you have any questions regarding this, I'd be happy to answer. So here ends my first chapter. Now the second chapter is also based on the bioelectronic platform. Now, if you remember, I told you the antibiotics act on certain vital components of bacteria. Now, of those vital components, one is the cell membrane. Now, usually the antibiotics which act on the cell membrane of the bacteria are not generally prescribed to any patient. These type of antibiotics are only given to patients who are on deathbed or are in critical care unit. And this happens because we have only 4 to 5 such antibiotics which target cell membrane of the bacteria. So to develop antibiotics from a wide array of molecules which target cell membrane of the bacteria, we need rapid screening and antibiotic discovery platform. So for this, what we did is we took out, instead of testing the antibiotics on the bacteria as a whole, what we did, we took out the bacterial cell membrane using a proprietary method wherein we passed the bacteria through 100 nanometer polycarbonate membrane, and the bacteria is chopped in this manner to form bacterial membrane vesicles. And these vesicles were then used to form, and these vesicles were then characterized to see if they are more relevant to the naturally produced vesicles as well as the synthetic vesicles of POPC. And we found that their structure and dispersity is similar to that of the naturally produced vesicles. And then we also found that their topography is similar to that of the bacterial membranes. We went further and checked their height as well as their thickness, which was found to be similar to that of the bacterial membranes. Now, what we did, we took the solution of these vesicles and uh, placed them on glass surface. We found that these vesicles can readily adhere to any surface uh, and form continuous supported lipid bilayers, which was characterized using SEM and AFM. Just to check if these supported lipid bilayers have same height, we scratched these lipid bilayers with a diamond cutter and saw that the thickness of these membranes is similar to that of the bacterial membrane. But further, to check if these membranes are fluidic and live, we performed FRAP, wherein we bleached the membrane with laser, high intensity laser, and let the lipids move towards the bleach area. And after some time, over a period of 360 seconds, we found that these lipids were able to move and reform the membrane into an intact one. And through this, we found that it forms a true non-static coating, which is fluidic and biomimetic. Now, these experiments were performed on glass to check if this mechanism is surface agnostic. What we did, we used technologically relevant substrates like P.PSS, graphene, and ITO, and placed the membrane's BMV solution on it, and it equally adhered to the surface of these technologically relevant surfaces. And we also saw not only on 2D surface, but this phenomenon was also equally transferable to 3D structures like gold nanoparticles. We could coat the gold nanoparticles with structures, membrane structures, as well as the optical fibers. This really shows that BMVs can be readily adhered to any relevant substrate, increasing their potential in better bioelectronic nanostructured devices and functional materials. But this is just one side. On the other side, to check if these membranes can really be used for antibiotic testing, what we did, we first did a control study wherein we treated the bacteria with polymyxin B, which is a membrane degrading antibiotic. And we saw when the bacteria was treated with polymyxin B, it poked hole in the bacterial membrane, thereby killing the bacteria. Now, when similar thing was done with these membranes formed on the glass, we saw that nearly identical pore was formed on these membranes. This shows that the system which works with the bacteria can also be transferred onto only membranes formed with uh, the bacterial membrane vesicles, which becomes a powerful and valid model for studying membrane and drug interaction. But now, again, this doesn't happen in real time, right? We are first treating the membrane with the antibiotic, membrane degrading antibiotic, and then we are doing AFM. It doesn't really reflect a real time study, as well as it is more tedious performing AFM. So what we did the same thing and standardized the protocol for SEM. Now, when the membranes formed were treated with membrane degrading antibiotics, we could see holes on the membrane, and the holes were gradually increasing based on the concentration of the antibiotic used. So this membrane disruption, we found that it is dose dependent, and it creates a quantitative screening tool using SEM. But again, SEM is also not a real-time tool, although it is more easier than doing AFM, but it is not a real-time tool. So we wanted to make it more quantitative and real-time. So what we did, we coated the working electrode with P.PSS, and we formed a BMV-derived bilayer on it. Then this method, how it works by measuring the membrane's electrical potential, which would be exactly higher, since membrane acts as an insulator and has higher resistance. So what we did, we checked the resistance of the membrane, then we treated the membrane with antibiotic damaging the membrane, which created pores for ions to pass through the membrane, resulting in the reduction of the resistance, allowing us to assess the effect of antibiotics in real time in a label-free manner, that too using just the EIS. So this was the setup. We took a chip wherein it had an electrode of 200 nanometers by 200 nanometer size. We formed a membrane on it. This membrane was then treated with polymyxin B. The membrane was degraded when it was treated with polymyxin B, and then we assess the membrane resistance before and after the treatment. So you can clearly see the membrane resistance is high when it is intact, then it reduces based on the concentration of antibiotic use. So we first performed this thing with Pseudomonas aeruginosa isolated membrane, and then we confirmed the same thing with Escherichia coli isolated membrane. So we tested two different bacteria and we got similar results, which demonstrates a powerful and quantitative label-free platform for testing antibiotics. Now, all in all, what we can do is we can use this method for high throughput testing. We can also advance this for better biosensors, which have receptors and efflux pumps from bacteria, all from mammalian cells, for better diagnosis. Then we can also extend this method for targeted drug delivery by BMV coated nanoparticles or BMV encapsulated cargo, or can also be used for fundamental biophysics studies, which we are doing at present. So for example, since we saw one type of model when we treated it with antibiotics, but on the same hand, we also saw different types of membrane architectures when they were treated with different types of antibiotics. And this is usually reported in the literature using just theoretical models. But this was the first time we saw it experimentally. So it can also be extended for fundamental biophysics studies. So all in all, this finishes the second chapter of using membrane-based bioelectronic model for high throughput screening of antibiotics. Now, I'll be focusing on biohybrid strategies for tackling AMR. So if you remember, I told you that bacteria develop certain mechanisms like enzymes, efflux pumps, which could throw out excess of antibiotics, making the bacteria more resistant. So one one way of increasing the efficiency of available antibiotics is blocking these resistance mechanisms. So over here, we focused on the resistance mechanism, which is efflux pumps. It is a pump which is responsible to throw out excess of antibiotic molecules from inside the cell to the outside. We did this study on Pseudomonas aeruginosa again, which has an efflux pump name as MEX-AB-OPRM, situated in the lipid bilayer. Now, this efflux pumps are ideally amenable to uh, a blockage by certain small molecules. The molecule that we used here as an adjuvant was vanillin, which is one of the most popular flavor ingredient. I'm sure everyone likes vanilla flavor. So imagine vanilla along with antibiotic, increasing the efficiency of antibiotic. This is something we wanted to explore. So we took vanillin, but the problem with vanillin is it is not soluble in water. So we had to make it more bioavailable. So the way we did it was using vanillin for the synthesis of gold nanoparticles, making a stable solution of vanillin capped gold nanoparticle through a simple method of green synthesis. The nanoparticles which we got were around 25 to 30 nanometers in size. Now these nanoparticles were used as an antibiotic adjuvant. So for example, if you want to use an antibiotic adjuvant to increase the efficiency of antibiotic, you need to show that this adjuvant itself does not act as an antibiotic. So we grew the bacteria in the presence of this vanillin capped gold nanoparticle, and we found that even at high concentration, as high as 2000 microgram per ml, these nanoparticles were not bactericidal. So we can take this as an antibiotic adjuvant. So what we did, we treated the bacteria with an antibiotic called meropenem. Uh, and to kill a particular amount of bacteria, we needed around 200 microgram per ml of meropenem antibiotic. But when we added vanillin capped gold nanoparticle, the amount of antibiotic needed to kill the same amount of bacteria was reduced by 10 folds. So you can clearly see the difference in the amount of antibiotic needed. So these adjuvants really work towards increasing the efficiency of the antibiotic. And this potentiation effect of vanillin capped gold nanoparticles were seen against two different antibiotics, which are usually also treated as last line antibiotics. So the third recommendations of WHO of reducing the amount of antibiotic was also achieved using the nanobiohybrid approach. And then we also saw that these antibiotics can Then we also saw that these vanillin capped gold nanoparticle can block the efflux pumps. So for example, these are bacterial cells which are streaked on a media containing ethidium bromide, which is a fluorescent dye. So what happens is ethidium bromide is also a substrate for efflux pumps. So when there is ethidium bromide, the bacteria throws out the efflux pumps and the bacteria does not flourish. But when we added vanillin capped gold nanoparticles in the media, the the gold nanoparticles blocked the efflux pumps, thereby retaining the ETBR inside the cell, and this ETBR then flourished inside the cell. So this assay clearly shows that vanillin capped gold nanoparticles are acting on the efflux pumps as an adjuvant and thereby increasing the potency of certain antibiotics. So this is one such food-safe molecule-based approach, which involves nanotechnology to increase the efficiency of antibiotics, thereby targeting certain resistance mechanism, especially the efflux pumps. So this is one of the strategy which targets the third recommendation of WHO. Now, the fourth recommendation we are working on is developing a nanomotor based approach to tackle the bacterial infection without really relying on antibiotics. So for example, what we did, we took bacterial membrane vesicles uh, through the same method, through extrusion made from E. coli. Then we coated these vesicles on magnetic nanoparticles. Now what happens is, when these magnetic nanoparticles are coated with bacterial membranes, these are not being recognized by the bacterial cells and they are readily uptaken. So what happens when these magnetic cores go inside the cell, we can use external magnetic field to either capture the bacteria or kill the bacteria by increasing the frequency of the magnetic field. So initially what we did, we built the magnetic core through solvothermal method. Then we disguised the magnetic core using bacterial membrane. And then we let the bacteria co-culture with this disguised core and let the bacteria internalize the bacterial internalize the nanomotor core, and then we use magnetic field to capture the bacteria. So this is the initial result wherein we developed the magnetic nanoparticles. The bacterial membrane vesicles produced through the similar method of extrusion. Then the coating was confirmed using ATIR, FTIR, wherein we got similar peaks with respect to the lipids as well as some of the proteins coated onto the magnetic nanometer. Then through fluorescent microscopy, we could assess that the magnetic nanomotor is getting attached to the bacteria. Similarly, it was confirmed through SEM. The bacterial membrane run parallel across the cell membrane of the bacteria as well as the nanomotor, which was also confirmed spectrophotometrically. Then what we did was we used magnetic field to capture the bacteria. Here you can see the captured bacteria, as well as use higher magnetic field to rupture the bacteria. So the magnetic core comes in outside rapidly, reducing the bacteria into the pore. So this is one of the strategies which does not really require antibiotics to kill the bacteria. And the one more strategy that we worked upon is coating the magnetic core with viruses. Now these viruses are bacteriophages which only target the bacteria and not humans. So their virus-based strategy is very easy to use against such bacterial infections. So what we did, we capped the magnetic nanomotors with such bacteriophages, and we used them to penetrate into the bacterial membranes, into the biofilms. Now the magnetic field helped the magnetic core to enter the bacterial biofilms and release the viruses into the bacterial cells, thereby killing the bacteria. And the efficiency was altogether more than that of virus and microbots alone. Now, this article is already published. You can scan and go through the article in detail. Now, this forms the fourth strategy, and the recommendation, it follows the recommendation of WHO. I would like to thank my mentors from my PhD through the course of my entire postdoc for guiding me through this amazing field, as well as my colleagues for helping me out to realize these ideas and concepts. Thank you so much.
Thank you so much, Sagar. That was super insightful talk and a lot of things happening there. Thank you so much again for your time
Thanks so
and
much.
presenting for us. So a kind reminder for everyone, if anyone has a question, it's the time now to ask. You can either type it in the Q&A box or raise your hand, and I will enable your mic to speak up. Okay, so people start typing. Maybe while they are typing, I can ask one question. I would like to ask you about the first chapter that you presented. Regarding like, I know you talk a lot about the device itself and the mechanism, but I'm curious about the backend side of how you connect in terms of microfluidics. like the connection to the inlet and the outlet, and which type of pumps, and does flow rates, pressure thing, all of these parameters affect the results that you have?
Yes, definitely. The flow rate and the pressure definitely affects the results. So we kept the flow rate constant to that of the urinary tract system because we wanted to take it further for urinary tract system studies.
Okay.
So these things really affect the measurement. That's what we found, and we generally use the peristaltic pumps to
Okay.
manage the flow. Yeah, to manage the flow and also to see if the biofilm is moving from one place to another.
Yeah. And do you couple it with some kind of a flow sensor to check like if the pump is pumping in what expect in the accepted range, or you just rely on the pump itself?
For now, we just relied on the pump itself.
Okay.
Yep.
Okay, good to know. Okay, there's another question from Ifran. She said, moving away from scientific technical challenges, how can insurers and to some extent governments insensitive biopharm to invest in R&D regarding the discovery, design, development, and distribution of these novel approaches, system platform, and antibiotics? Did you
Okay, so
get,
this is
yeah, okay,
yeah,
cool.
this is more sort of administrative thing. I mean, a
Yeah,
lot of players come into this picture because it's not about only the academicians who are going to going to work in this area. There has to be a coordinated effort from maybe pharma, academia, industry, as well as the government. The main thing is altogether, it's our task to make the government aware about this issue. So, of all the recommendations that I mentioned about WHO, the first recommendation of WHO is awareness. If there is no awareness created by academicians like us, there'll be hardly any coordinated R&D effort towards the discovery and development of such type of platforms. That's what I feel.
okay, thank you, Sagar. Uh, if there is there any question? If anyone has any question, please type it or raise your hand. And yeah, if there is no more, we can maybe close up the session for today. Thank you very much again, Sagar. Thank you for answering the question and
Thanks
for
so
the
much,
nice
Latifa.
seminar. Thanks a lot for your time. So we will have our next seminars in the next 3 weeks. I'm going to send an email for everyone who registered in this seminar about the next speaker and link for registration. And until then, I would say we'll catch you later by that time. And I would say thank you again, Sagar, and yeah, we'll see you hopefully soon.
Sure. Thanks so much, Latifa.
Okay. Thank you so much. See you everyone
Yeah.
in 3 weeks. Bye.